The principles of genetics. What is DNA? DNA stands for deoxyribonucleic acid. It is a long molecule that contains each person's unique genetic code. It holds the instructions for building the protein that are essential for our bodies to function. DNA instructions are passed from parent to child, with roughly half of child's DNA originating from the father and half from the mother. It is a two-stranded molecule that appears twisted, giving it a unique shape referred to as the double helix. Each of the two strands is a long sequence of nucleotides or individual units made up of phosphate molecule, a sugar molecule called deoxyribose containing 5 carbons, and a nitrogen containing region. There are 4 types of nitrogen containing regions called bases, namely adenine, thymine, guanine, and cytosine. The order of these 4 bases form the genetic code, which is our instructions for life. The bases of the 2 strands of DNA are stuck together to create a ladder-like shape. Within the ladder, adenine always sticks to thymine and guanine always sticks to cytosine to create the rung. The length of the ladder is formed by the sugar and the phosphate groups. Here are the step-by-step process of DNA replication. First is the preparation for replication. Before DNA can be replicated, the double-stranded molecule must be unzipped into 2 single strands. DNA has 4 bases called adenine, thymine, cytosine, and guanine that form pairs between the 2 strands. The interactions between base pairs must be broken by an enzyme known as DNA helicase. The replication fork is bidirectional. One strand is oriented in the 3-inch to 5-inch direction, which is called the leading strand, while the other is oriented to 5-inch to 3 inches lagging strand. The 2 sides are replicated with different processes to accommodate the directional difference. Step 2 is where the replication begins. The leading strand is the simplest to replicate. Once the DNA have been separated, a short piece of RNA called a primer binds to the 3-inch end of the strand. The primer always binds as a starting point for replication. Primers are generated by the enzyme DNA primase. Third step is the DNA replication called elongation. Enzymes known as DNA polymerases are responsible for creating the new strand by a process called elongation. DNA polymerase III binds the strand at the site of the primer and begins adding new base pairs complementary to the strand during replication. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria such as E. coli, polymerase III is the main replication enzyme, while polymerase I, II, IV, and V are responsible for error checking and repair. In eukaryotic cells, polymerases alpha, delta, and epsilon are the primary polymerase involved in DNA replication. The process of replication is discontinuous as the new created fragments are disjointed. Lastly is the termination. An enzyme called exonuclease removes all RNA primers from the original strands. Telomeres act as protective caps at the end of chromosomes to prevent nearby chromosomes from fusing. DNA polymerase can only add nucleotides in the 5' to 3" direction. So basically, at the end, replication produces two DNA molecules, each with one strand from the parent molecule and one new strand. The parent strand and its complementary DNA strand coalesce into the familiar double helix shape.